Today we will learn about different types of point mutations in DNA and their effect on proteins. I will explain these mutations using an analogy where I equate a warehouse's operations to the operations of a prokaryote. My father manages a warehouse as a family business, which is why I chose this analogy. We can see a DNA template strand transcribed from right to left into an mRNA that encodes for a protein containing 5 amino acids. From this point on, we will only deal with the coding strand to showcase any point mutations. Here is the general warehouse analogy. A company placing an order is like interface in the DNA replication cycle because the money prepares the order to take place. My father is like the DNA polymerase because he creates a replication of the company's order. Gail is like the RNA polymerase because she photocopies the order, with the photocopy being like an mRNA strand. Mitch is like the ribosome because he reads the photocopy, while Peter is like the tRNA because he brings the packages. The pallet is the protein because it is stacked with packages similar to how a protein is made with amino acids. Here, Carson makes a mistake similar to a silent mutation. He copies the order as package 345 instead of 344, which results in the wrong package added to the pallet. The customer won't know the difference though because the package contains the same items, so the pallet is overall unaffected. A silent point mutation is shown in the coding strand when adenine is replaced with cytosine, which results in a change in the mRNA sequence. However, the AGG and CGG codon encode for the same amino acid, so the protein is overall unaffected. Adenine being changed to cytosine is an example of transversion because adenine is a purine and cytosine is a pyrimidine. Here, Karsten makes a mistake similar to a missense mutation. He copies the order as package 354 instead of 344, which results in the wrong package being added to the pallet. The packages contain different items, so the pallet contains one wrong box, and the customer does not get all of what they ordered. A missense point mutation is shown in the coding string. Guanine is replaced with cytosine, which results in a change in the amino acid sequence. As a result of the change, a 309 amino acid is encoded instead of an arginine amino acid. The protein now has one different amino acid, which may change its function or make it non-functional. Guanine being changed to cytosine is an example of transversion because the nucleotide ring structure changes from purine to pyrimidine. Here, Karsten makes a mistake similar to a nonsense mutation. He copies the order as package 304 instead of 344. Package 304 does not exist, so Peter does not bring the package to the pallet. Mitch moves on to the next order to save time, so the pallet order does not end up being fulfilled. A nonsense mutation is shown when adenine within the DNA sequence that encodes for the lysine amino acid codon is changed to thymine. This is because thymine is changed to uracil when creating mRNA, and the resulting amino acid sequence, UAG, is a stop codon. The resulting program loses the lysine amino acid and is now one amino acid short. This protein may have different functions or be non-functional altogether. Adenine being changed to thymine is an example of transversion because adenine is a purine and thymine is a pyrimidine. Here, Karsten makes a mistake similar to a read-through mutation. He copies the order as package 101 instead of 100. Usually, Peter knows an order is finished after 100 packages. However, Peter brings package 101 and then even more packages to the pallet due to the mistake. The resulting pallet has more packages than the customer ordered. A read-through mutation is shown here when the thymine within the coding strand is changed to adenine. This results in an amino acid sequence that encodes for lysine instead of a stop codon. The result is that the protein continues to be transcribed and includes more amino acids than it should. The protein will most likely not perform its original function. Thymine being changed to adenine is an example of transversion because thymine is a pyrimidine while adenine is a purine. I hope you enjoyed this video and learned about how when my dad makes mistakes at work, it is kind of like a DNA point mutation.